Hello, everyone. Welcome to today's session. I am Ankita. I work as a sales and marketing director for Medchem Express for the Australia and New Zealand region, and I will be your host for today. I hope you all are excited, as excited as I am, to share the insights about antimalarial drug targets and therapeutics in this area. It is my pleasure to welcome our guest speaker for today, Dr. Brad Sleebs. He heads the laboratory in the New Medicine and Diagnostics Division at the Walter and Eliza Hall Institute of Medical Research, Melbourne. Dr. Sleeves earned his PhD from La Trobe University and began his research career at WeHi in 2005. Over the ensuing years, he has established himself as a leading medicinal chemist and chemical biologist. His past research includes the development of anxiolytics in collaboration with bionomics and agents that target the BH3 family of proteins for the treatment of blood cancers in collaboration with Genentech and AbbVie. His current research focuses on developing small molecule probes to better understand biological processes that are essential to the survival of parasites that impact both human and animal health. In parallel, Dr. Sleeve leads drug discovery programs to design new therapeutics to treat parasitic diseases in collaboration with academic and industry partners, including Janssen Pharmaceuticals, Medicines for Malaria Venture, and MSD. We at Medchem Express are proud to have been able to support Brad's research along the way. So specifically, it's rewarding for us to have him here. And before we start, just a quick note, we have allocated 15 minutes after the talk for discussion, but please feel free to type in any questions you have during the presentation using the Q&A feature here. Please join me in giving a warm welcome to Dr. Brad Sleeves again, and I'd like to give it over to Dr. Sleeves for the main presentation. Over to you.
Wonderful. Thanks, Ankita. I will just share my screen. Hoping that you can see the screen.
Yes, we can.
Yeah. Wonderful. Thank you. Thank you for the kind introduction. And today I'm going to tell you about some of my research that was conducted at the Walter Eliza Hall Institute here in Melbourne, Australia. And so some of the research that I'll tell you about today is in the optimization of antimalarial probes that we use to better understand disease pathogenesis of this devastating disease known as malaria. But we also generate these antimalarials by way of hopefully creating antimalarial therapeutics that can benefit those that are suffering from this disease in endemic areas. So I'll just start with a bit of background behind the disease, malaria. It's caused by 6 Plasmodium species that are known to infect humans. P. falciparum is the most deadly as it causes up to 70% of all known cases and up to 90% of all known deaths. P. falciparum is almost exclusively endemic to sub-Saharan Africa, but is also found in Southeast Asia and also the Central Americas. And P. falciparum is also the most readily cultured in a laboratory setting, and will be the focus of the talk here today. Quite unacceptably, this disease, malaria, still causes close to 600,000 deaths annually worldwide. And so why is this disease so difficult to treat? One of the reasons is that this organism, Plasmodium, has a complex multi-stage life cycle, and it's rather cunning in that it has found ways that it has evolved ways to be able to evade the host immune system. So with the life cycle, there are two hosts, the mosquito host and the human host, and the parasite is transmitted from one human host to another when a female Anopheles mosquito is infected with the organism, takes a blood meal on the human host. It then injects a form of the parasite known as the sporozoite, which then enters the circulatory system and makes its way to the liver. Here in the liver, it invades a hepatocyte cell where it self-propagates thousands of copies of itself before exiting that hepatocyte and re-entering the circulatory system. Here in the form of the mirror, as in the form of a myrizoite, it then invades a red blood cell, and this is what initiates the asexual blood stage. And here in the asexual blood stage, there's many replicative cycles of this stage, which the parasite both exits and re-enters the red blood cell or other blood cells, and in doing so destroys many red blood cells. And this leaves the host feeling rather anemic, having flu-like symptoms, and this is why we call this stage the symptomatic blood stage. At one point in this blood stage, the parasite generates a male and female form of itself known as the gametocyte, and this is the form of the parasite that's transmitted back to the mosquito, which then goes on to infect another human host. So historically, the treatments for malaria are generally listed here on the top left of the screen, and they are rather a narrow subset of chemotypes that have been used over many years. And I draw your attention to the timeline down below, indicating the time at which the antimalarial was introduced to the field. And in the black arrow is the time of which resistance was first detected against each of these antimalarials. And as you can tell, the time from when it was introduced to the time resistance was first detected is a relatively short period of time. With that in mind, around the turn of the century, the World Health Organization then suggested that we should be using these antimalarials in combination with one another to try and mitigate the onset of resistance. And this has helped somewhat, and the combination therapies that are used as frontline therapies now are known as artemisinin combination therapies. And as you can see, since their introduction, there has been resistance detected against artemisinin, which is rather unfortunate, but it's just a mutation in a protein that doesn't actually make these combination therapies ineffective. It just makes them have a slight delay to efficacy. And so for most part, these combination therapies are still very effective. So why do we want to develop new therapies if we still have effective treatments? Well, we want to future-proof now for when these therapies become ineffective due to resistance. So at the turn of the century, there was a call put out by World Health governing bodies for academia and industry to join forces to mass screen small molecule libraries against the malaria parasite to uncover novel starting points for the development of new antimalarials. From this initiative came these molecules first that are shown on the screen here. And as you can tell, they have chemical structures that are diverse from the historic treatments that are being used to treat malaria. These compounds also have novel mechanisms of action, and they also target multiple stages of the parasite's life cycle. Unfortunately, like most drugs going into clinical trials, the attrition rate of these compounds or antimalarials is just as high. And so at least two of these compounds have dropped out of clinical trials due to toxicity and pharmacokinetic issues. Additionally, in patients that have been treated with these compounds, they've extracted parasites that have resistant mutations. And that is concerning, given that this is only in clinical trials, and you can only imagine what would happen once these molecules move into the field. So, with the current issues in clinical trials and the attrition rate and the issue with resistance that may impact the effectiveness of current antimalarials, there's still an ongoing need to develop new antimalarial therapeutics to future-proof against resistance in the attrition rate in the clinic. So what attributes should an antimalarial compound have? Here I'm showing the target candidate profile, which is of a single antimalarial agent. And this is typically closely aligned with the activity that they have across the parasite's life cycle. So, for example, if we wanted to create a prophylaxis treatment, ideally we want to treat the liver stage, which would prevent the establishment of the symptomatic blood stage infection. If we wanted to obviously create a treatment, we would obviously need a compound that would reduce the asexual parasitemia load, reducing the symptoms in the patient. And ideally, if we would create a therapy that was rolled out in malaria endemic areas, we would want to target the transmission stage to block the transmission of the disease in that endemic area. We have a look at the target product profiles. Which is a combination of two of these or more of these antimalarial agents. And there's two of these target product profiles, a single exposure radical cure and a single exposure chemoprotection. I'd like to draw your attention to the wording of these target product profiles, in particular single exposure, which means one dose cure. And that's quite a high bar to set to treat any disease, let alone malaria. But that's the challenge. So what attributes do we have to build into our antimalarials that we're developing to have an oral single exposure cure? These compounds must have high absorption and systemic exposure, a long half-life systemically. This is so there's enough compound on board and for long enough to be able to clear parasitemia effectively. These compounds must be fast acting, and if they're not fast acting, then they should be partnered with a fast acting compound. This is to be able to clear parasitemia as quickly as possible in these patients. The compounds must have a high barrier to resistance and no cross-resistance to known clinical resistant strains. And obviously safety is paramount with any drug, but more so with an antimalarial drug. Safety in children and pregnant women is also important, given that that population cohort is most affected by this disease. So I'm going to give you just a small snapshot of what we've been doing with our antimalarial program today. In particular, with our collaboration with Janssen Pharmaceuticals, working with the Medicines for Malaria venture. And in the second part of the seminar, some of our work with the Burnett Institute here in Melbourne, Australia. So for the first part of the seminar, I'll focus on the collaboration with Janssen and MMV. And this collaboration really was initiated when we here at WeHi took delivery of the Jumpstarter library, which is an 80,000 small molecule library that have that are structurally diverse and are drug-like. And so we screened this library against the P. falciparum asexual stage parasite, using a lactate dehydrogenase readout for parasite growth. Using this screening cascade, which included a screen of confirmed hits against the HepG2 human cell line, just to make sure that the compounds weren't broadly cytotoxic. We arrived at a number of confirmed hits that fell into a variety of different structural series, of which the highest ranked structural series and at least exemplars of them, those series are shown on the screen here with their asexual EC50 values. So what I thought I'd do today is just give you a flavour of what we've done with most of these chemical series by concentrating on this tricyclic series here. And so the aims of this research were medchem optimization of the antimalarial activity while monitoring and improving in vitro ADME properties to get to a point where we could have a suitable molecule go into a mouse model to show proof of principle efficacy, to establish the resistance profile to ensure these compounds had a high barrier to resistance or otherwise, to identify the mechanism of action and explore the parasite biology, and to phenotypically characterize the compounds across the parasites, or the compound's activity across the parasite's life cycle to be in line with the target candidate profile that I mentioned earlier. So first with the medchem optimization, and here's just one exemplar from this structural series shown here with its asexual stage EC50 value and the activity against the human HepG2 cell line. Just to note that this structure does have a chiral center in it at this carbon bridgehead. And for most part, throughout our structure activity relationship study, we reduced analogues in a mixture of R and S stereoisomers because we didn't have a stereospecific synthesis to generate either isomer. But I'll come back to separation of these isomers a little later on in the talk. But to start off with our medchem optimization, typically we want to look at each point of the molecule that is synthetically viable to access and determine whether or not those modifications are either adventitious or deleterious to antimalarial activity. And so I'll just give you a brief snapshot of how we optimized this chemical series. So firstly, looking at the substitution on this nitrogen, we found that smaller substituents such as the methyl group were most ideal. Looking at the exocyclic ring system, we found that a chloro substituent in the 3 position of this arrow ring, but other small substituents were also equally as tolerated. And those 2 iterations gave us a 3-fold boost in activity. We then looked at substitution on this arrow ring here, and what we found in the 7 position was that not only a chloro, but a variety of both bulky and small substituents, both polar and non-polar, were tolerated in this position. And this gave us another 5-fold boost in activity, taking the activity down to the realms of what we see in this assay with the drug chloroquine. And so it's not all about optimizing the antimalarial activity. We want to get to the point of dosing into a mouse model or moving further into the clinic. We also have to consider other properties such as metabolism. And so here we have the turnover of this compound in the presence of liver microsomes and hepatocytes. And showing here is the intrinsic clearance values, which are very high, meaning our compound's being rapidly turned over. And ideally, we want Clint values less than 10. The aqueous solubility at physiological pH of this compound series was also low. Ideally, we want to be above the 50 micromolar mark. We also look at parameters such as this permeability across a CACO2 cell monolayer, which is an indicative prediction of intestinal absorption. So here we're going from apical in one chamber across that CACO2 monolayer to basal lateral in the other chamber, and we're in a pretty good position here. And going back the other way, we can get an indication of whether or not this compound is a substrate for drug transporters, and we're in a relatively good position for that as well. We also screened our compound against a panel of high-risk targets, which can give adverse of pharmacology moving forward into the clinic. And one notorious target is the cardiac erg ion channel, for which we don't inhibit at all. And so what we wanted to do was improve on our metabolism and solubility moving forward. To do this, we decided to incorporate more polarity into our molecule, particularly in this exocyclic ring system where we observed oxidative metabolism by MET-ID. And we found that an endocyclic nitrogen in this position here maintained antiparasitic activity and started to improve metabolic stability and also aqueous solubility. We then incorporated a polar heterocycle into this 7 position here, and also a fluorine into the 5 position. And this maintained, again, antiparasitic activity while improving metabolic stability and markedly improving aqueous solubility. We finally then separated out the stereoisomers at that carbon bridgehead and found that the S isomer was the active constituent, and we were able to do that by chiral chromatography and determine the absolute configuration of the stereochemistry here by X-ray crystallography. And as you can see, we're in a reasonable position for metabolism and also aqueous solubility. However, we have to be conscious of the metabolic stability in mouse liver microsomes when we're moving into a mouse model. And that's what we did. We moved into a mouse model, and the mouse model we used was known as a P. falciparum humanized skid mouse model. And the reason why we're using humanized mice here is because P. falciparum is a human infectious organism. It doesn't infect mice usually. And so to be able to infect mice with P. falciparum, we continually engraft human erythrocytes into these mice before we infect them with the asexual stage P. falciparum parasite. Then we allow parasitemia to build up, and then we dose our compound orally from days 1 to 4 and measure parasitemia across those days and out to day 30. And if we have no parasitemia at day 30, this is denoted as a curative dose. And what we observed with our compound, what we observed in this model, I should say, in black here, is increasing parasitemia over time with the vehicle control. But with our compound, at an oral dose of 25 mg per kg, we reduced parasitemia to below detectable levels. And this was 1 day quicker than chloroquine at a slightly lower dose. However, we did see parasitemia recrudesce after day 8. So obviously we didn't completely clear parasitemia in this model. And perhaps we thought we may have done a little bit better with this molecule, provided the attributes that it does have. So what was the reason behind that? When we looked at the plasma exposure levels in mice of this compound, we found that the plasma exposure levels were lower than we really originally anticipated. And the reason for that was that we attributed it to was the low intestinal absorption, and perhaps now this compound had become a drug transporter substrate also. So if we were to further develop this series, we'd have to go back and improve on those parameters. So moving on to establishing the resistance profile and identifying and exploring the mechanism of action of this compound series. In my mind, the most successful way to uncover the mechanism of action is using forward genetics. And here what we do is we treat parasites in culture with compound for a prolonged duration of time. And over time, these parasites slowly become resistant to the compound. Then we extract the genomic DNA of those resistant parasites and whole genome sequence that DNA to detect the genetic aberrations responsible for the resistance. An iteration of this workflow, which is known as the minimum inoculant of resistance assay, which is a platform run by David Fiddig's lab at the University of Columbia, is very similar, but the only difference is that we're using a range of parasite inoculum or different levels of parasitemia typically in a 96 well plate, and then we measure for recrudescence for up to 60 days. And this allows us to detect the rate of onset of resistance. And so we subjected this early frontrunner compound W714 to this assay platform, and we detected a number of recrudescing wells from days 13 to 60. We then went on to perform dose response of the first recrudescing wells, and you can see here the EC50 shift between the DD2 wild type line and the recrudescent populations here. And so taking these details of this assay together, this places our compound in the medium risk category. Ideally, we want to be in the low risk category. And being in this medium risk category was one of the factors that precluded our compound from further development. Nonetheless, we then took these recrudescent populations and performed whole genome sequencing, and what we detected in two populations was a mutation in each of those populations in a gene known as ATP4, and in another population, amplification in the gene ATP4. So what is ATP4? It's an ATPase sodium channel, which is essential for maintaining sodium homeostasis in the parasite cytosol. It's also a proton antiporter, so it's also important for maintaining pH in the parasite cytosol. PF-ATP4 is a known and established antimalarial drug target, with these two compounds here advancing to human clinical trials. At the time we're doing this research, there wasn't a cryo or M structure of PF-ATP4, but there now is. It's been recently published in Nature Communications. However, at the time, we were relying on using a homology model, which was based on this rabbit calcium channel for which there was an X-ray crystal structure. And we were able to map our resistant mutations from our studies to this homology model, along with the mutations detected to these two clinical candidates. And you can see here that they cluster closely together, which signifies perhaps our compound and these two clinical candidates may bind in a conserved binding site around these mutations. We wanted a little bit more evidence. We were targeting PF-ATP4, so we then performed some cross-resistance studies with these two clinical candidates against our resistant clones. And as you can see here, versus the DD2 parental line, we can see an EC50-fold shift with these resistant clones with the two clinical candidates. Conversely, we then tested our compounds against the resistant mutations, or parasites, should I say, that were detected and generated with these two clinical candidates. So, for example, this G358S mutation in PF-ATP4 was detected in parasites that were taken from patients in human clinical trials that have been treated with K609. And you can see how ineffective these two compounds are against this mutant strain. But that wasn't the case against with our compound series that had very subtle reduction in activity against this mutation. And this is one reason why we and others were quite excited by our chemical series and had an interest in developing it initially. Nonetheless, we tested our compound against this triple mutant PF-ATP4 mutant, and as you can see, we have a 5-fold 10 decrease with our compounds against this mutant, which was similar to the clinical candidates. So this gave us some more evidence that we're targeting PF-ATP4 with this compound series, but we wanted more of a phenotypic readout. And so we turned to Adele Leanne at the Australian National University. It has this specialised assay, which I'll walk you through. And so they extract the parasite from the red blood cell, and then we load these parasites with this sodium-sensitive diet. And then they add in our test compound in the DMSO control. And then they measure fluorescence over time as a surrogate for sodium in the parasite cytosol. And so what we can see from this assay is both the clinical candidate K609 and our active isomer both increase sodium levels in the parasite cytosol, but not so our less active enantiomer, which has a similar effect to the DMSO control. So this signifies that we're inhibiting p-fatty-before in parasites and increasing the sodium concentration in the parasite cytosol. With this PFADBase assay, we can also perform this in a dose response with the active and less active enantiomer, and these EC50 values very much reflect the EC50 values we see in the parasite growth assay. We can also perform this assay with the G358S mutant, where we have a subtle increase in EC50 with this compound here. And again, these ADPase values roughly are in line with the values that we see in the parasite growth assay. So all in all, this is very strong evidence that indeed we are targeting PFAD-before. So moving on to phenotypically characterizing the activity of these compounds across the parasite life cycle, I've shown you the activity of these compounds against the asexual blood stage. What we do know is that these compounds don't have, these compounds, PFAD-before inhibitors, don't have any activity across the liver stage, and that was indeed the case with our compounds. But PFAD-before compounds are known to have activity in the transmission stage, so we wanted to confirm that with our compounds. And one way to do that is this dual gamete formation assay that's been run by Michael Dell's lab initially at Dundee University, where they take stage 5 gametocytes, they add in the test compound or the DMSO control, and then they induce gametogenesis by the addition of xanthorenic acid or a drop in temperature. And this mimics the mosquito midgut conditions. And then we look for male gamete viability by quantifying moving flagellating centers, and this is done by an imaging algorithm. And for females, viability is quantified by a female-specific marker. And what we saw in this assay from our initial hit compound right through to our front-runner compound were increasing potency against both male and female gametes. And just to note that the front-runner compound had very similar activity to the clinical candidate. So moving on to a mosquito model, and the mosquito model we're using here is a standard membrane feeding assay where we take, again, late stage gametocyte infected blood that has been treated with our compound or the DMSO control, and we feed this infected blood to these mosquitoes through this membranous device here. And then we allow parasitemia to mature within these mosquitoes for up to 7 days. And then we dissect the mosquito midgut and count the oocysts in the mosquito midgut by microscopy. And what we observed with the DMSO control was a significant oocyst burden per each mosquito midgut, but this was significantly reduced at 50 and 100 nanomolar potency with our compound, at least the active isomer of our compound, but not the less active isomer, which had a very similar burden to the DMSO control. So this signifies that indeed our compounds could be used in a transmission blocking therapy. So just in brief summary of this first part of the talk, we've taken a hit from a high throughput screen. We've been able to optimize the antimalarial activity, improve ADME properties to get to a point where we have proof of principle efficacy in a mouse model of malaria, or a malaria mouse model, but future improvement, particularly in the absorption, will be needed if we were to further develop these compounds. For genetics, identified PFAD-before as the molecular target, which was validated by drug resistance profiling and further phenotypic assays. However, the risk of resistance of this chemical class really precluded it from further development. And although the resistance profile was akin to other PFAD-before inhibitors, we still believe that PFAD-before inhibitors with a high barrier to resistance do indeed have a potential in a curative or a transmission control antimalarial therapy. So hopefully that just gives you a flavour of some of the work that we've been doing with all these chemical series, for which we've now optimized them all apart from one chemical series, which was refractory to optimization. We've also optimized all the, identified the mechanism of all these chemical classes. Now, in some instances, the mechanism wasn't the actual molecular target, it was a mechanism of resistance. So, for example, this ABCI drug transporter. In other instances, we identified known drug targets such as cytochrome B, which is the drug target of atovaquone. And in other instances, we identified new targets that weren't previously described, such as this start lipid transfer protein. And so we started to publish on this work if you want to go and further read about it. So next, I'll just move into the next part of the seminar, which is our long-standing collaboration with Paul Gilson's lab at the Burnet Institute in Melbourne, Australia. And so Paul Gilson's labs had a long-standing interest in identifying compounds that inhibit invasion, but also egress of the parasite out of the red blood cell. And he has a specialized assay that is able to read out these events. And in doing so, he's been able to use this assay to screen a number of boutique libraries. And here's just some of the compound classes that we've worked on together over a number of years. And again, we've also optimized these compounds with respect to the antimalarial activity, and we've also identified the mechanism of action of these compounds. In some instances, we were picked to publication by other groups in terms of their mechanism. And again, we've started to publish on some of this work, but today I'm just going to give you a flavour of what we've been doing with Paul's lab by concentrating on this MMV512 compound. And then again, the aims of this research were very similar to the PF84 research in terms of showing you the medchem optimization, establishing the resistance profile, and identifying and exploring the mechanism of action. So just to give you a very brief snapshot of the key points of modification to optimize this chemical class, we started with this MMV512 compound, and we're able to substitute in these two N-methyl groups, which gave us a significant boost in anti-parasitic activity. We then substituted in this isopropyl group here, which gave us another 5-fold boost in activity. And many different iterations around these L rings enable us to have compounds such as WHI-264 with single-digit nanomolar activity. And so again, it's not all about optimizing the antimalarial activity. There's other parameters to consider, such as metabolism and aqueous solubility. And here you can see from the starting point to our front runner compounds, we haven't improved on the metabolic stability, nor have we improved on the aqueous solubility. And so this is really did preclude this compound class from further development toward the clinic. But nonetheless, we're still able to use these compounds as chemical probes to explore, hopefully, some novel parasite biology. And so that's what we did next, is to go ahead with 4Genetics to establish the resistance profile and identify the mechanism of action. And again, just to remind you, we're using 4Genetics here, in which we generate resistant parasites by treating parasites with our compound for a long duration of time. And then we perform whole genome sequencing to detect the genetic aberrations responsible for that resistance. And this was conducted at Paul Gilson's lab, where he generated a number of resistant populations to MMV512. And you can see here the marked EC50 shift between the wild type parental line in grey and the populations in these different colours here. Whole genome sequencing of those resistant populations, at least in 2 of the populations, revealed mutation in the rhomboid protease 8 and in another population in the CSC1 transporter. We also performed minimal inoculant resistance in collaboration with David Fiddick's lab at the University of Columbia. Again, here we do this to determine the rate of resistance, and it's done with a range of parasite inoculum, typically in a 96-well plate over 60 days. And again, we detected a number of recrudescing wells from day 14 to 53. Here you can see the EC50 fold shifts between the parental line and the first recrudescing populations. Taking these parameters together, this places our compound class, and again, in the medium risk category, and this would have been another factor that precluded this compound from further development, as we would have liked to see it be in the low risk category. But nonetheless, we performed whole genome sequencing on these parasite populations and detected a mutation in 2 of the populations in the CSC1 transporter, and in another population, a mutation in the rhomboid protease 8. So what is CSC1? It's a cation permeable stress-gated channel belonging to a family of osmotic sensitive cation channels. And it's got this really neat mechanism by which it has this cytosolic domain, which senses cations in the cytosol, and any perturbation in that cation concentration brings about a conformational change in the cytosolic domain, which is transferred to these pore-forming helices, which allow in the influx of cations to correct the dysregulation of cations in the cytosol. So CSC1 is one of many transporters expressed in P. falciparum, and it's essential by a mutagenesis screen in asexual stage parasites. But the role of CSC1 and P. falciparum asexual parasites is completely unknown. So we created a homology model of P. falciparum CSC1 based on this Arabidopsis channel, for which there was a X-ray crystal structure, and we mapped the mutations to this homology model, and they localized to this region of the protein, which perhaps signifies that our compound could bind to this region of the protein. So what is rhomboid 8 protease? Well, it belongs to a family of intramembrane serine proteases. It's one of 10 rhomboid proteases expressed in P. falciparum. Rhomboid 4, 6, 7, and 8 are essential for asexual parasite development, but the role of Rom8 in P. falciparum is completely unknown. Again, we created a homology model of this P. falciparum rhomboid 8 protein based on an X-ray crystal structure of this E. coli rhomboid protease. And again, we mapped our mutations to this protein, which clustered around this substrate binding cleft of the protein, and noting that the catalytic residues are located at the bottom of that cleft. So perhaps our compound would bind in this substrate cleft of this protein. So what are the roles of CSC1 and ROM8 in P. falciparum development? Does CSC1 function as a cation transporter? And what is the functional relationship between CSC1 and ROM8? And these are just some of the questions we wanted to answer with these proteins. But first of all, to answer these questions, we really first of all concentrated on working on CSC1, starting by generating a conditional glucosamine knockdown of CSC1 in parasites, which was generated by Titania Conning-Ward's lab at Deakin University in Melbourne, Australia. And so to do so, just showing you the construct here. So we introduced this targeting construct into the endogenous locus of CSC1, and the construct contained a triple HA tag, a glutamate ribosome switch, and a selectable human DHFR marker. And this was introduced by CRISPR-Cas9 technology. And just showing you the PCR here, showing the correct integration of that construct. Here we're showing you on addition of glucosamine to these parasites, we have an approximate knockdown to 30% of the original quantity of CSC1 versus this control here. On the addition of the glucosamine here, we can see that we're knocking down parasite growth over a number of asexual stage cycles versus normal wild type line here. And here we're showing you that on increasing addition of glucosamine, we have decreasing activity of our small molecule MMV512. against asexual stage parasites. And below here, given we've got a HA tag, we've performed immunofluorescence microscopy to show that this protein localizes to the parasite cytosol. So what cations does CSC1 transport or translocate? We're pretty confident, and data I'm not showing here with the lack of time, that this transporter doesn't transport calcium or potassium, but what on the evidence I'm going to show you, we're pretty convinced that it does transport sodium. So again, turning to this assay that's run by Adele Arn's lab at the Australian National University, where we isolate parasites from the host erythrocyte, load them with sodium-sensitive dye, and add our test compound as a surrogate for sodium in the parasite cytosol. So here, what I'm showing you is that we're using the clinical candidate K609, which is a PF80B4 inhibitor at 50 nanomolar concentration, which is the super maximal concentration where we see 100% inhibition of PF80B4 in parasites. And we're also dosing it with our compound WEHI-264 at a relatively high concentration. And as you can see, the increase in sodium in the parasite cytosol is very similar to the supermaximal concentration of K609, but not our inactive control compound, which has no effect on this assay and similar to the VMSO control. So how can we delineate between inhibition of PF80B4 and CSC1 with this assay? So one of the assays that we did, experiments we did run, was to dose our compound in combination with K609. And what we see in this instance is we see a doubling effect of the sodium concentration in the parasite cytosol up above the supermaximal concentration of K609. So here we're inhibiting PF80B4 and we're also modulating the effect of CSC1 to get that doubling effect of sodium concentration in the parasite cytosol. We can also run this assay with a glucosamine knockdown CSC1 line. So here we're showing you no knockdown, so no addition of glucosamine, showing a nice dose response with our compound up to the supermaximal concentration of K609. But here we're knocking down CSC1 in these parasites in this assay, and we have a blunting effect with our compound. We can also run this assay using the mutant CSC1 line and the ROM8 mutant line detected in our resistant studies. And what we're showing you here is the effect on the parental line, so effectively a dose response up to the supermaximal concentration of K609 again, but again with our mutant CSC1 line and the ROM8 mutant line, we see a blunting effect of this response. So our working model at the moment is that we believe that PF80B4 is the master regulator of sodium in the parasite cytosol, and that CSC1 is acting as a surveillance protein, keeping PF80B4 and the sodium concentration in check. And what we believe ROM8 is doing is assisting in regulating CSC1 in terms of its function. And just to note that our CSC1 knockdown and our mutant CSC1 and ROM8 parasite lines at a basal level have no effect on the resting sodium concentration. So, but when we add our inhibitor, what we believe is happening is that by some inhibition mechanism that our compound is inhibiting ROM8, which is dysregulating its effect on CSC1, allowing the influx of sodium into the parasite cytosol. And we believe this is what's leading to parasite death on the addition of this compound class. So there's many questions that remain still with this biology, and those are the questions we're trying to answer at the moment. How does ROM8 regulate CSC1? Well, in mammalian systems, there's some evidence for a rhomboid protease regulating a calcium ion channel of the same class. How does our compound series bind to ROM8? Well, again, in mammalian systems, there is evidence of these lactam-like small molecules that irreversibly bind to the catalytic serine of these rhomboid proteases to inhibit their function. And it's probably no coincidence that our compound class also contains a lactam. So it's possible that our compound class has the same inhibitory mechanism. And does CSC1 function like a dimer as many of these mechanoosmotic cation channels function? And we've drawn a molecular model of this, and we believe it's quite possible that CSC1 could also act as a dimer. But really, we do need a structural biology to express these proteins and define the further biology of these proteins to answer these questions, as well as create a rhomboid 8 knockdown parasite line to assist also. So very brief summary of this research that I've shown you here. We've been able to optimize the antimalarial activity of this compound class to produce potent probe compounds, but the in vitro ADME really did preclude this compound class from further antimalarial development. 4 genetics identified rhomboid 8 and CSC1 as appear to be target of this compound class, which was confirmed by reverse genetics and phenotypic assays. And we believe that these two protein function together to regulate sodium levels in the parasite cytosol. And again, structural biology will really be key to further defining the role of CSC1 and ROM8 in parasite development, as well as a ROM8 parasite knockdown line, which we're currently working towards. And that just leaves me to thank the following people on the screen, which have collaborated to not only the work I presented today, but the work on all the molecular series that are presented during the presentation. And those names highlighted in bold have been particularly helpful in the research that I presented today. And I'll leave it there, and I thank you for your time and attention.
Thank you, Brad, for that wonderful presentation. Let's open the floor to questions. I would encourage everyone to post your questions here in the Q&A feature using the Q&A feature, as I mentioned earlier. Meanwhile, Brad, I just had a question. I can see that you've used some structurally diverse drug-like compounds, the 80,000 compounds that you started with. What are the opportunities you see around repurposing or reoptimizing some existing compounds, classes for malaria or other parasitic diseases?
Yeah, sure. So I suppose I'll base the answer to the question on antimalarial research. So I didn't mention earlier, but the Janssen 80,000 compound library was a vendor comprised library. So they did have molecules that we could go and access from screening vendors. But for most part now, because of this great push to screen both academic libraries and vendor comprised libraries, over the last 20 years, effectively the whole vendor comprised or commercially available small molecules have been screened against a malaria parasite. And for most part, this data is freely available online at the ChemMBEL database for people to go and search and look at and follow up on if they so wish to do so. But so in effect, screening other vendor comprised libraries is probably not of any value anymore. And so getting back to your point, Anika, with screening repurposed compounds, in my opinion now, I think that the only way to uncover really new starting materials and new chemical matter to embark on development of antimalarials is to screen these hidden compound repositories at pharmaceutical companies and academic libraries that have never before been screened against the malaria parasite. And so And as evidence of this, we've had a long-standing collaboration with MSD in America and have been able to discover a novel target class that we've now brought into human clinical trials. So I think that's the way forward for the future of antimalarial research at the moment.
Great. Thank you. Thanks, Brad, for answering that. So if there's no other question, I'd like to sneak in one more, which is the question is rather broad. You built this amazing industrial partnership for this project. How important do you think, such partnership, academic industrial partnership is, how critical they are to sustain innovation in an area like malaria? Which is relatively underfunded, if I may so, compared to other research areas.
Yeah, so at least at our institute, the Walter and Eliza Hall Institute, we don't, like many academic institutes, don't have the expertise or the resources to take a small molecule from early discovery phase right up into the preclinical, into clinical trials. And so for that aspect, we must partner with industry, or a pharmaceutical company does have that expertise. And MNV, Medicines Malaria Venture, have been one of the world leaders that do have that type of expertise to be able to advance compounds into human clinical trials. So collaborating with either a pharmaceutical company or, for example, the Medicines Malaria Venture do have the expertise and resources to do that. For academia is absolutely critical. At the stage of which you partner or start collaborating with industry or Medicines Malaria Venture, just depends on a variety of different factors. But in my mind, the earlier you um, start establishing the collaboration with the partner, I think it's more beneficial for both the academic partner and also the industry partner. And it probably also expedites the transition towards the clinic, doing that as early as possible as well.
Sure. Thank you. Okay, I can see no other questions posed here. But yeah, if you have any questions, please feel free to get in touch with Brad. And if there's any compounds, products that has come to your mind throughout the presentation, come get in touch with us at Medchem Express. We do have quite relevant antimalarial products and libraries, also virtual screening libraries as well as service side of capacities for such kind of research projects. So yeah, we'll be happy to get in touch and provide our support. Please stay tuned to attend our upcoming webinars. And I'd like to thank Dr. Brad Sleebs again for your precious time today and everyone for joining in. And see you all in our next webinar then. Have a good day, good evening, wherever you're based. Thank you.